A cell culture production process involves two stages, a cell propagation stage, followed by the product formation stage. The cell propagation stage may include a series of microcarrier cultures with increasing reactor volumes before it reaches the production stage. In these seed reactors, one should avoid maintaining cells at a confluent stage over an extended period. In general, it is preferable to use a culture at its late exponential or subconfluent stage of growth. Furthermore, cells at the subconfluent stage are relatively easier to be trypsinized from the microcarriers than those cells at the confluent or post-confluent stage. The first step in detaching cells is the removal of the medium and the washing of cells. The microcarriers are allowed to settle, and the medium is removed by suction. The washing step can be carried out either in a spinner flask or in a centrifuge tube. Compared to roller bottles or tea flasks, the washing step for microcarriers has to be much more extensive. Because of the high porosity of the dextran beads, a large amount of medium is retained inside the beads, even after extensive washing. Furthermore, the cells clinging to the dextran beads slow down the diffusion of medium components from inside the beads to the bulk liquid. The cell-laden microcarriers are resuspended in the washing buffer and maintained in suspension by agitation for a short period before they are allowed to settle again, and the supernatant is withdrawn. The effect of washing will be readily visible by the decreased intensity of the phenol red color in the microcarrier beads. Multiple steps of washing are usually needed with the use of 30 to 50 volumes of washing buffer. The steps following the initial washing vary with cell strains and with microcarriers. For a number of cells grown on Cytodex 1 or controlled charged microcarriers, after the initial washing, cells are ready to be exposed to trypsin solution for detachment. However, for other cells, especially those grown on gelatin-coated microcarriers, another incubation step is often needed to facilitate initial morphological change before cells are exposed to trypsin solution. A balanced salt solution without calcium and magnesium, containing a small amount of glucose, is used in the incubation stage in our laboratory. To this solution, a 0.5 millimolar of EDTA is also included. The incubation can be carried out at 37 degrees Celsius. The incubation time ranges from 10 to 20 minutes. During the incubation period, cells begin to round up and change from an elongated shape to a more spherical shape. This incubation step is thought to shorten the time of the subsequent trypsin treatment and facilitate the cell detachment. How trypsinization is carried out is dependent on the pH of trypsin treatment and on whether it is necessary to remove trypsin afterwards. If a sufficient dilution can be achieved in the subsequent cultivation stage and the pH is neutral, then the trypsinization can be carried out in the original flask or a test tube. Before the trypsin solution is added to the microcarrier, the liquid volume of the microcarrier suspension is reduced to 2 to 3 times that of the settled microcarrier beads. Trypsin solution is then added to give rise to the desired concentration. In some cases, the removal of residual trypsin is critical, such as when serum-free medium is employed. There are also cases when a higher pH, in the range of 8 to 9, is used in the trypsinization, especially when controlled charge microcarriers are used. The buffers used in the washing and incubation steps have a neutral pH. For high pH treatment, it is necessary to replace the residual buffer with the high pH trypsin solution. For both removal of residual trypsin and high pH treatment, the use of a chromatographic column or other filtration device is convenient. The cell-laden microcarriers are mixed with the trypsin solution, poured into the filtration device, and allowed to settle. The trypsin solution is then drained from the bottom of the microcarrier bed or withdrawn from the top by suction. The trypsinization step takes only a very short period, ranging from 30 seconds to a few minutes. During exposure to trypsin, cells develop a spherical shape. Fresh medium is then added to stop the proteolytic reaction. Cells are suspended in the medium, and mild shear force is applied in many different forms. Mild pipetting is the easiest way to provide shear force in a laboratory. In our laboratory, we have found it convenient to pass the cell microcarrier suspension through a column packed with glass beads. The trypsinized microcarrier and cell suspension can be passed through the column by gravity. Because of the high void volume, the passage through the column is rapid, and the amount of microcarriers or cells retained on the packed glass beads is relatively small. The column is further washed with fresh medium. Microscopic examination should be performed to ensure that most of the cells are detached from the microcarriers. In general, about 95% of cells can be detached, with a viability of 90 to 95%. Some cells reattach rapidly to the microcarriers from which they were detached. Therefore, it is advisable to maintain the temperature relatively low to decrease the reattachment rate. In the cases where a high multiplication ratio is used, as in the cultivation of vero cells and many other continuous cell lines, the cell microcarrier suspension can be inoculated directly into a new culture. On the other hand, if a low multiplication ratio is used, as in the cultivation of human foreskin cells, FS4, a large amount of used microcarriers may be carried over to the new culture. To separate cells from the microcarriers from which they were detached, avoid passing cells through a packed bed of used microcarriers, such as in the case of dead-end filtration. For a rapid separation of cells from used microcarriers, a tangential flow-type filtration should be used. The filter is made of stainless steel screen with 60-micron openings. In the course of filtration, the microcarriers are collected at the bottom of the filter, while the free cells readily pass through the cylindrical screen at the center and are collected at the bottom. The subsequent inoculation steps are essentially the same as those for the inoculation from the roller bottles. Cells trypsinized from a seed culture readily reattach to a new microcarrier culture after inoculation.